Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speakers. We'll also have just a few questions for you before the Q&A, so we'd appreciate your participation when the time comes. So thank you for joining us today. It's now my pleasure to introduce our speakers, Ellie Zackham, Senior Manager, Process Development from Rooster Bio, Lauren Torres, Field Application Scientist, and Jeremy Neidert, Bioprocessing Account Manager, both from Repligen.
Good morning, everyone, and thank you for joining our webinar today. My name is Ellie Zackham. I am the Senior Manager of Process Development at Rooster Bio. In today's webinar, I will be joined by Lauren Torres and Jeremy Neidert from Repligen. First, let's go over the goals of this collaboration, what challenges we are overcoming, and highlight what we wanted to achieve. There has been a growing demand for extracellular vesicles, or EV supply. This is due to their emerging role as intracellular messengers and due to their therapeutic potential as targeted and natural delivery vehicles with high specificity and efficiency. The number of clinical trials investigating mesenchymal stem cell derived EV as a therapeutic has been greatly increasing over the years. In order to meet the need, the field needs to industrialize and intensify functional EV manufacturing processes. EV manufacturing comes with challenges. Given the high complexity and fragility of the EVs or exosomes, our goal is to choose the right fit platforms to increase recovery, maintain bioactivity, and reduce variability. The challenge of scaling up the manufacturing of complex products like EVs necessitate well-designed upstream and downstream platforms to ensure consistent and reproducible yields. To address those challenges, Rooster Bio partnered with Repligen to develop robust, scalable manufacturing processes for EV therapies that are affordable and readily accessible to patients using Rooster Bio's industrialized EV production platform and Repligen's bioprocessing solutions with scalable and low-shear technologies. To learn more about this collaboration, please refer to the press release referenced at the bottom of the slide. The paradigm for EV manufacturing is summarized in this workflow. EVs were produced using Rooster Bio's high-quality mesenchymal stem cells, media system, including growth and EV collection media, bioprocesses for EV production, along with Rooster Bio's powerful analytical tools. Processing of the EVs was performed using Repligen's clarification, TFDF technology, and ultrafiltration and difiltration, KTF technology. I will start by covering the work accomplished by Rooster Bio, and I will be followed by the Repligen team to cover the downstream part. I would like to first start by introducing Rooster Bio. Rooster Bio was first launched in 2013 with the goal to fill the gap in supply chain for mesenchymal stem cell-based products by standardizing and industrializing the supply chain with the products that we launch. We design our unique manufacturing processes for MSCs and EVs that are flexible and scalable that anyone can access by simply purchasing our products. Since the focus of today's webinar is developing manufacturing processes for EVs or exosomes, I will walk you through the concept of EVs. EVs are defined as particles produced or released by cells. EVs range in size from 40 up to 1,000 nanometer. EV is a general term which includes exosomes, microvesicles, apoptotic bodies, among others. As part of their composition, EVs contain proteins, nucleic acids like RNA and DNA, and lipids, which are considered as cargoes that EVs deliver to recipient cells and are thought to have an important role in the therapeutic impact of EVs. Additionally, EVs contain transmembrane proteins, which are on the surface of the EVs and are used as identity markers. Why is there interest in EVs and specifically MSC-derived EVs? MSC EVs have similar regenerative capacities as MSCs. That's why MSC EVs are so popular. They make cell therapy easier because your product is no longer living cells that you have to keep alive. It is a secreted, complicated molecule with better stability and more straightforward engineering and an easier regulatory profile. So, how does Rooster Bio help standardize and industrialize EV manufacturing supply chain? It's by the products that we launch. Our products are high volume cell banks, which are the working cell banks within the context of the manufacturing process. Highly efficient paired xeno-free expansion media that comes with bioprocess recommendations to rapidly enable customers to get into scalable bioreactor-based production processes for bulk MSC expansion and EV production. We also have low particulate xeno-free EV production media that allows for increased EV yields and ensures clean EV collection without having to worry about non-EV contaminants. We also have CGMP versions of many of our products. Our goal is to help our customers generate billions of MSCs and therefore trillions of EVs from the bulk cell mass. I will now discuss the study's flow diagram to generate EVs. We used xeno-free rooster vial bone marrow MSCs that we cultured in a C train, followed by their inoculation into a stir tank bioreactor at a 3-liter scale. EVs were generated following established critical process parameters that Rooster Bio has developed. Briefly, cells were grown in Rooster Nourish for 5 days without the need of any media exchange. At the end of the growth phase, growth media was removed, the cells were washed, and Rooster Collect EV media was supplemented. EV collection was performed over 5 days. At the end of the collection, the EV conditioned media was harvested from the bioreactor for further processing. Now I will hand it over to Lauren, who will cover the first part of the downstream processing.
Thank you, Ellie. Hello, everyone. Thank you for joining us today. I'd like to dive into Repligen's contribution to this collaboration. If you're not familiar with Repligen, we specialize in bioprocessing solutions. We have a global presence across the world with offices and manufacturing sites in the following locations. Here are some examples of the technologies we offer, which span upstream, downstream, and analytical solutions. You might be familiar with some of the systems, services, and products that we offer. Today, I'll be focusing on process intensification systems. Shown here again is our workflow. My portion of the collaboration focuses on cross-flow TFDF technology, which was used for clarification of the EV material. This system was selected because it was able to address multiple challenges. In the next few slides, I'll be going over how this system enabled a faster, scalable, sterile solution for this process. In case you're not familiar with the cross-flow TFDF, it is a system that was launched in 2019. TFDF stands for tangential flow depth filtration, as it combines the technologies of tangential flow and depth filtration into one technology. So as you can see there, it has a depth filter-like membrane encased in a hollow fiber-like casing. The pore size of the depth filter-like channels are 2 to 5 micron, which are great for large molecule processing. The larger pore also allows us to pass the full population of EVs through to the permeate stream. The filter comes pre-assembled in a gamma-irradiated flow path, which allows us for plug-and-play operation. Since we're able to run in an enclosed sterile filter, we're also able to skip a sterile filtration step and pass the permeate material straight on to downstream for further processing. For the development of this process, we did several phases of studies to identify process parameters. In phase 1, we split a 3-liter bioreactor into 3 aliquots to screen conditions using a 3 centimeter square fold path. Ultimately, we selected the following condition based on flux, recirculation, and pressure profiles. In phase 2, we scaled up the process to a 30 centimeter square flow path. And in phase 3, we experimented with different processing vessels as well as demonstrated reproducibility. For this process, we achieved a turbidity reduction of 60.3%, which allowed us to go straight onto downstream TFF processing. We also achieved a recovery yield of 81%, which was comparable to the centrifugation control. And ultimately, this technology allowed us to reduce processing time risk, and will ultimately be able to scale up to a 240-liter bioreactor scale, which is Rooster's desired manufacturing scale, as well as be under 2-hour processing time with a 2100 centimeter square flow path. Now I'll pass it on to Jeremy to explain the UFDF portion.
Thanks, Lauren. Hi, my name is Jeremy Neidert, and I perform this work as an application scientist at Repligen, just like Lauren. I'll be reviewing the UFDF development of this collaboration. So through screening and scaling experiments, we developed a concentration and diafiltration TFF step. This process was designed to be scaled up using Repligen's KTF large-scale system and hollow fiber filters in order to provide a low shear solution for better product quality and recovery. So for the TFF development side of things, we actually started with a sample of the TFDF clarified material. And because of the low turbidity of the sample we had coming straight out of TFDF, we eliminated any normal flow depth filtration in this process and went straight into TFF. So in phase 1 of the TFF development, we screened various molecular weight cutoffs of hollow fiber filters to understand the retention. We screened 300, 500, and 750 kilodalton, with the understanding that the highest molecular weight cutoff that would retain the sample could also provide the best purification that would let through some of the smaller impurities and provide a higher flux. So in phase 2, now that we had found our critical process parameters, we did a small scale study to lock down the process with those parameters. And then in phase 3, we scaled up the process to accommodate the full volume being processed in each batch and provide scale-up reproducibility and confirmation data. For all of these studies, we used the Repligen KR2i benchtop TFF system, which provided us the flexibility for the range of these scales of various studies, as well as the walkaway automation to provide the robust control of our critical process parameters and our reproducibility studies. So in phase 1, first we quantified the initial material coming out of TFDF to understand the particle sizing and concentration. We took a sample through our permeate of each of the molecular weight cutoffs, and we ran that same analysis to understand the concentration of what size particles were making their way through the filters and where you did not want them. The primary starting material had species starting around that 136 and above nanometers. So we found that even with our highest molecular weight cutoff tested, 750 kilodalton, we saw very limited product in the permeate and retained over 99% of our product in the retentate, which confirmed the use of the 750 kilodalton molecular weight cutoff. During this initial screening, we ran flux excursion studies for each of these molecular weight cutoffs to understand this retention, as well as develop the TMP that we would be running the process at. We ran each molecular weight cutoff at a few different shear rates and various TMPs to optimize the flux. So when we found our ideal transmission or ideal molecular weight cutoff, we had these parameters we could move forward with. So using the molecular weight cutoff, TMP, and shear rates found in those initial screening experiments, we ran a 600 mil small scale study at the same loading in order to lock down these parameters in a full process run. We found a processing time of less than 3 hours. In this run, we saw a typical flux decay and pressure profile similar to most UFDF processes with no filter fouling. We also confirmed total recovery above 90% to this step with less than 1% of our EVs found in the permeate. This provided us with the confidence to move forward to the next stage of the development. So with these same process parameters, we then moved to process the entire 4-liter batch through 3 reproducibility runs. The volume varied a bit based on the upstream batch volume, but each of these runs saw similar performance across all 3 reproducibility runs in terms of flux, pressure profile, and processing time. Across all 3 of these runs, we saw favorable recovery and again confirmed less than 1% of permeate breakthrough, confirming this high recovery of the product and reproducibility of this process. So lastly, using the data from our small scale study and our scale up and reproducibility runs, we are confidently providing the scale up solutions for Rooster's current scale up goals using Repligen's KTF large scale system and the cross-flow max hollow fibers. So with that, I'm going to pass this back to Ellie to review the analytics used in this process to allow us to come to some of these conclusions. Thank you.
Thank you, Lauren and Jeremy. In addition to EV recovery data, we looked at different critical quality attributes or CQAs of EVs. At Rooster Bio, we have developed different EV analytical testing based on guidelines provided by the Minimal Information for Studies of Extracellular Vesicles, or MISAV. We offer testing for EV identity, purity, and bioactivity. As part of identity, generated particles in this process were stained with a membrane dye that is specific to lipid bilayers. Positive stain with the membrane dye indicates intact lipid bilayer, which is a characteristic of EVs. The percent of positive membrane staining did not change in TFDF or TFF samples compared to harvest samples, confirming that the particles in this process are EVs and were maintained during processing. Some of the impurities found during the generation of EV conditioned media include proteins, DNA, and RNA. We measured the concentration of those impurities after processing with TFF and compared their content to harvest samples. We saw over 50% clearance, indicating that TFF was successful at clearing some of those impurities. We further confirmed the identity of those EVs through Western blot by staining the particles with surface markers or tetraspanins, which are considered as identity markers for EVs. Those tetraspanins include CD9, CD63, and CD81. Each blot contains 3 samples. Lane 1 is the harvest sample, lane 2 is the TFDF sample, and lane 3 is the TFF sample. The presence of bands in each lane for each marker confirmed the identity of the EVs and their maintenance during processing. In addition to confirming the identity, purity, and composition of the EVs, we also evaluated their bioactivity using an in vitro wound closure assay. Cells were grown as monolayers in wells, followed by the creation of a gap. The cells were then treated with the EVs generated from this process. Closure of the gaps were observed and quantified and reported as percent wound closure in comparison to negative control. Results have shown that EVs generated in this process have closed over 80% of the created wound, which was significantly higher than the negative control, which was close to 40%. This confirms that those EVs are functional after being processed. Those results show that the processed EVs not only maintain their identity, but also maintain their biofunctionality. In summary, we hope we have shown you today that Rooster Bio's highly productive upstream processing platform produced potent MSC EVs that were incorporated into Repligen's downstream processing platform. Results in this work demonstrated the successful development of a simplified MSC EV downstream process with TFDF and TFF by eliminating the need of a depth filtration step. Through this work, we have identified scalable parameters for large-scale processing. High recovery yield was consistently achieved through the TFDF and TFF unit operations. The process was successfully completed in a short processing time at all scales, which was completed in less than 3 hours. Advanced EV analytics demonstrated that processed EVs met the minimal information for studies of extracellular vesicles or MISAF criteria and were potent in an in vitro wound closure assay. And lastly, I would like to thank both the Rooster Bio team and Repligen team who contributed to the success of this work. And with this, we're happy to take any questions.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speakers now. But before we get to the Q&A, we just have some questions for you, the audience. You should see them pop up on your screen now. The questions are, where in the bioprocess workflow are you working? What volume of scale are you working or targeting? And what molecules are you currently working with? So if you could just take a few moments to answer those few questions, your participation would be greatly appreciated. And then after, we will get into the Q&A. We'll just take a few moments here. See some answers coming in already. And once we begin the Q&A, you should still be able to answer these questions. All right. Well, fantastic. I see a lot of good answers coming through. If you haven't answered yet and still want to, you still can. We appreciate all your participation. Um, so with that, I think it's good we go into the Q&A now. We see some questions coming in. Our first question is, were the cells preconditioned before generating EVs?
Hi, yeah, I can answer that question. My name is Steven Lenzini. I'm a process development scientist at Rooster Bio. I had to step in for Ellie Zackham. So, yeah, as we showed in the process flow diagram, there's after inoculation in the bioreactor, there's a growth phase where the cells expand and reach a confluence level where then we exchange the growth media with our EV collection media, Rooster Collect EV. This is a media that's designed to be low particulate and not contain any components from the growth media so that the EVs are in a relatively pure state before going into downstream processing. Now, our process is fully compatible with, I know there are some groups that want to initiate some kind of treatment of the cells before initiating EV production, such as priming with cytokines or other treatments. And our process is fully compatible with this kind of decision. So that could be done as well. But in this study, we didn't do any priming or preconditioning other than just our media Rooster Collect.
All right, and our next question is, are you using any kind of permeate flux control on your final TFF step?
Hi, this is Jeremy Neidert from the Repligen side. I can answer that one. So we did not use any sort of permeate flux control. We didn't need it for this experiment. We just did a shear control of 4,000 per second shear, as well as a 5 psi TMP that was found through our flux excursion. Thanks.
And our next question is, was biocompatibility performed on custom consumables per ISO 10993? If so, how extensive was it? How many parts of the standard were tested to?
I can field this question. This is Lauren Torres. Specifically for the TFDF flow paths, they do undergo rigorous biocompatibility testing. They undergo USP Class 6 testing to show no long-term mal effects. Thank you.
And similarly with the TFF flow pads, we have similar testing as well. We can provide at Repligen fully customized gamma irradiated flow pads ready for GMP processing.
And what is the EV storage buffer and temperature for long-term storage?
Hi, this is Steven again. So we didn't really explore the formulation of EVs in this study, although this is something that Rooster Bio is very interested in. Typically, looking at the literature and some of our own internal testing recommends minus 80 C storage for purified EVs. And I think in this study we used phosphate buffered saline, which is a commonly used buffer for storage of EVs.
Our next question is, can you speak to TFDF membrane lifetime or fouling?
Hi, this is Lauren again. So TFDF filter fouling is highly process dependent. However, higher recirculation definitely helps with increasing the lifetime of your filter. So it depends on what kind of recirculation values that you'll be required to run it. The main priority is for higher recirculations, which causes a sweeping motion to prevent particles from laying down on the membrane.
And our next question is, the impurities clearance seems to not be efficient, does your process meet criteria or purity? What is the remained protein impurity?
We do think that this process, although it works very well, will eventually be combined with then chromatography steps downstream, which will continue to purify the protein impurities from the conditioned medium.
And do you recommend to use the 750 kDa more than the 500 kDa cutoff to recover EVs? Do you have comparison of size distribution for recovery between both filtration?
Yeah, so we did screening on 300, 500, and 750 kilodalton. Because even with the 750 kilodalton, the largest we used, we saw really full retention of our EVs. We went forward with that in order to provide a higher flux, as well as provide more, passing of smaller molecules in there as well, and impurities. So we did test each one of them. We did flux excursions for each one of those as well. So that way, as soon as we had that clearance data, or that rather retention data, we then already had our flux that we are going to be moving forward with.
And our next question is, did you do a single collection of EVs after 5 days in Rooster Collect? And did you monitor nutrient consumption?
Yeah, so our process is designed at this point to do a single batch collection of EVs, and this is just to avoid, complicated batch definition. So it is a batch process, and it was a single collection on day 5. We routinely monitor nutrient consumption over time. If you're interested in this kind of data, we can provide this at Rooster Bio and point you to the right place to find it on our website.
And what chromatography mediums were used for affinity and IEX steps?
So similarly to Stephen's answers about impurities, this study did not include any chromatography steps. This is just the clarification with TFDF and the UFDF process with the hollow fiber TFF membrane.
And where do you prepare the source of your, I'm sorry if I pronounced this wrong, mesenchymal stem cells, and where are they isolated and processed for commercialization?
So we, at Rooster Bio, we provide cells and media. There are 2 tissue types that predominantly that we provide coming from either the bone marrow or the umbilical cord.
All right, well, it looks like we're running just a bit short on time, so if we don't get to your question, we'll be passing them along to our speakers who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, what is your intended use of EVs clinically? For example, IVIM or any other route?
Rooster Bio, we consider ourselves a provider of raw materials, and so we have many customers that intend to use EVs clinically in different settings. This would be a question better suited for them, although we want to do our best to develop the processes to be compatible with many different intended clinical uses.
Okay, fantastic. Well, I want to say thank you to Stephen, Lauren, Jeremy, and Ellie for a great presentation and Q&A session. And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with the direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.